Good morning, everyone. Welcome to the first webinar in ICA's new series, Navigating IC BPS, Conversations with Experts. Today's webinar will feature Dr. David Klumpp presenting bacteria as pelvic pain triggers and potential therapies. We're so excited you're here with us today. Just a few housekeeping items before we start. Please note that this session will be recorded. To ask a general medical question, not personal, please use the raise hand feature in Zoom. You will be asked to unmute yourself. Please stay muted when you're not speaking. You can also ask a question using the Q&A feature. Dr. Klumpp will answer as many questions as he can in our hour-long webinar. Please prepare your questions to ask Dr. Klumpp immediately following his presentation. Without further ado, please welcome Dr. Klumpp.
Well, thank you, Jenlene, and thank you all for joining us this morning. First, I guess a bit of a disclaimer here about conversation. So I'm going to lay kind of a biological groundwork or framework for our discussion that'll kind of come after this presentation. So I plan to speak for about 40 minutes or so, and then I'm happy to answer any questions after that. So I am David Klumpp. I'm the first thing to emphasize is that I am a basic scientist. I'm a researcher. I am not a clinician. So specific questions about your own health would be difficult for me to answer, and I'm not qualified to do so. So just keep that in mind. Nonetheless, I'm in the Department of Urology. I'm at Northwestern University in Chicago, where the weather is improving. First, about this pretty picture, or at least I find this picture pretty. This is from one of our mouse models of interstitial cystitis, or IC. And it is a tissue section, or a bologna slice, if you will, of a bladder from a mouse in one of our IC models. And this is what's called a fluorescent microscopy image. And just to kind of give you a sense of what we're looking at here, this blue dye is staining, it's called DAPI, and it's staining the nuclei of the different cells in the bladder. The red stain here is for uroplakins. These are proteins that are produced at high levels by cells lining the bladder lumen, the interior space of the bladder. And then these green cells here are cells that are actually dying. And you can see there's a loss of the continuity of this normal red layer here because a number of these cells have died. And this is something that we believe it mimics what may be happening in the IC bladder. All right, so a quick disclosure. I'm on the board of directors for the ICA. I'm proud to have joined in October. I do have IP relating to IC, but not yet any commercial entities, although we're working to address that. So IC, what is IC? Well, many of you are far more familiar with this than I am. My mother was a longtime sufferer of IC, so I know just how devastating that can be. The underlying biology, as you probably know, is not well understood, but there are a number of features of IC. This is a cystoscopic image. Many of you have probably seen these of a patient undergoing hydrodistension, and we can see these petechial hemorrhages, as they're called, where bladder is leaching, or where blood is leaching into the lumen or the center of the bladder. So that's what this image is. IC, as we know, is best characterized for severe and even debilitating pelvic pain. Voiding frequency, profound voiding dysfunction is also characteristic of IC. Comorbid depression is often associated with it. That can be linked to, and I'll define this term later, but HPA axis dysfunction, so altered stress responses and cortisol levels. There's often an association with mast cells. In my mom's case, I know that she's had in biopsies elevated levels of mast cells that were observed. There's damage to the urothelium being that tissue layer that lines the bladder, those red cells that I showed you a moment ago. And then there's some suggestion of altered levels, of vascular endothelial growth factor and that signaling. Probably all of you are familiar with the effect of comestibles or foodstuffs and how they can have impacts on the symptoms. Often acidic foods are known to, in many patients, cause issues and exacerbate symptoms, perhaps causing flares, as well as caffeinated foodstuffs. There's also there have been a number of publications of a history of UTIs in patients, and we'll see some examples of how that biology may come about. And then more recently, there's a sense that there's an altered microbiome, particularly in the gut of IC patients. All right, I'll be talking about that. So a little bit about innervation of the bladder. The bladder obviously is a storage organ, right? And you need coordination between relaxation of the sphincter, the urinary sphincters, and contractions of the smooth muscle of the bladder. And this is mediated by circuits in the spinal cord. But it's not just at the spinal cord. There are actually features up in the brain, the so-called pontine micturition center. So higher order places in the brain that regulate this. And one of the early models that we started using in our rodent studies, we did it in mice. It was originally shown by Luke Jasmine that if you inject rodents with pseudorabies virus, and this is a lot like zoster virus, the chickenpox virus that also causes shingles. If you inject mice with this virus, that it results in an IC-like condition of the bladder, at least histologically. And it turned out why we thought this was a very interesting model as opposed to others that might be employed in the field was that these effects on bladder inflammation that occurred as a result of this virus infection required intact upper urinary control circuits, such as the Barrington's nucleus. So we thought this was a good model. Now, you've all probably heard of the IC diet and the effects of these comestibles on aggravating symptoms. Why is that? Well, it turns out there's a lot of crosstalk between the bladder and the bowel. And this is actually a summary of data from the early 80s by McMahon and Morrison, where they were recording from cats, and they were doing spinal recordings from neurons in the sensory region of the spinal cord. And they were looking at responses to stimulating the bowel and the bladder. And they found that about a third of the neurons they recorded from up here received inputs from both the bladder and the bowel. All right. So that might be the basis for a lot of the effects of foodstuffs or the diet on IC, because you have this convergence of innervation from or signals from these pelvic organs at the spinal cord. And as you go higher up the CNS, the central nervous system, this convergence approaches about 75%. So it can even be difficult to discern pain emanating from one of these organs. All right, so many of you have heard of Dr. Theoharides. All right, he has been a longtime proponent for the role of mast cells, the mast cells in IC. And mast cells, many of you have probably heard of mast cells from a different context. These are often associated with allergic responses. So these are cells that release histamine, all right, and cause a lot of the hay fever symptoms if they're activated by a certain class of antibodies. But in this model here, what's been known for a long time is that mast cells can be closely associated in the bladder with certain types of sensory nerve fibers. And mast cells can respond to a stimulus called substance P that can be released from these. All right, this neurotransmitter substance P can activate mast cells, and mast cells, when they're activated, they release a wide variety of factors. one of which is histamine, which was thought to be able to feed back onto nerve cells. All right. But they also release a variety of pro-inflammatory mediators upon activation of mast cells that can cause inflammation in the bladder. And we started this by doing some very early experiments, just taking the soup coming off of activated mast cells and putting it onto urothelial cells in culture. And we found that a certain molecule, tumor necrosis factor alpha, caused this inflammatory response of urothelial cells. And TNF, you may have heard of this. There are a lot of medications that are advertised on TV, such as Enbrel and Humira, that target TNF functions. All right. Well, my first graduate student did a lot of work on TNF, and he was looking at mast cells and how they traffic, and actually showed that it was mast cells producing a little tickle of TNF and then more TNF as they were toward the urothelium, as they migrated toward the urothelium, and that caused the dying of cells in the bladder wall, like I showed you in that very opening fluorescence micrograph. All right. Now, that was before we started working on pain. So our group then started to look at pain responses in our various IC mice. And the first question you might ask is, how do you measure pain in a mouse? And we have a couple tests. One is very simple and one is a little more challenging. One thing we do is we measure, what's called tactile allodynia. And that is a response to stimulating the skin by poking with nylon monofilaments of different stiffnesses. You're applying a force to the skin. Now, why does that work? Well, it turns out that visceral organs like the bladder don't have a lot of pain fibers. All right. And what they do, they often share with a corresponding skin surface. And a classic example here is the perception of a heart attack often first being perceived as pain in the left hand or arm. All right. So the skin becoming sensitive. Turns out for the mouse, the corresponding skin region to the bladder is the pelvic region. So we poke the mouse a number of times with these fibers, and we see whether they become more sensitive, more responsive to this, what's called referred pain. The other slightly more challenging test for pain, and I'll show you a little bit of data from these systems. I don't want to overwhelm you with data at all, but we can actually distend the mouse bladder. Think of your own bladder filling. We use saline, and then if the bladder is very sensitive, that will cause a tightening of the abdominal musculature, this so-called vesicomotor reflex, or VMR. So we measure VMR or allodynia, are two tests for pain in mice. So using these systems, we were first looking at the mast cells, and we noticed that there was a difference that if we block the effects of TNF, that these mice had normal pain. So that told us that although TNF was causing damage to the bladder wall, it really wasn't causing the pain response. Right? So that led us to looking elsewhere, and we thought about, well, maybe the histamine is causing the pain. And indeed, that was the case, that it was mast cell histamine, and there are 4 different histamine receptors, and we identified 2 of them as being important in our system, histamine receptor 1 and then H2R. And in fact, H2R probably playing a more important role in our system. Okay, so it shows that mast cells can do a couple different things in bladder pathology and pain, and that these are separable events. Well, that kind of sets the stage for our work in bacteria and how they can contribute to pain. All right. And it kind of came from a simple question, because at the time, the other half of the lab worked on urinary tract infections. And urinary tract infections are most commonly associated with infection by uropathogenic E. coli, UPEC. All right. And so when UPEC are in the bladder, there's a robust inflammatory response. All right. And then there's discomfort. All right. A pain response. But there are also many individuals, particularly the elderly, and this in long-term care facilities can approach 100% of the residents can have their bladders fully colonized with E. coli. And this is a condition called asymptomatic bacteriuria, ASB. In an ASB, these individuals have bladders full of E. coli. They have robust inflammatory responses, but for some reason have no discomfort from that. Hence the asymptomatic aspect. All right. So we were interested in the difference between these two. We have a bunch of E. coli in the bladder. We have inflammation that's comparable, but very different symptomatic responses. We looked at a number of different factors and then eventually identified LPS as the factor mediating this pain response. So what is LPS? Well, LPS stands for lipopolysaccharide. That's why we just call it LPS, because that's a mouthful. And this is a glycolipid that resides in the, if we have our bacterial cell here, gram-negative bacteria actually have two membranes. And in the outer leaflet of the outer membrane, the dominant glycolipid is this molecule LPS. And this is a schematic of it. There's a lipid like a fatty tail called lipid A. There are a couple other sugars in here, and then there's a large carbohydrate structure, hence the glyco of the glycolipid. All right, so this carbohydrate structure can be quite large. It's composed of repeating units of a few sugars, from 2 to 6 sugars, which can have different patterns of branching, different composition. But this repeating unit gives rise to many different types, serotypes as we call them, of E. coli, over 180. All right. The function of this O antigen is not really known what it does, although I will show you one thing that it does in a few minutes. All right, so we found that lipopolysaccharide, when we purified them from our 2 different bacteria, causes very different things when we put it into the bladder of a mouse. All right, so here in red is LPS that's purified from bacteria, UPEC, that cause UTIs. And we see we get a very rapid increase in this allodynia, or measure of pain. All right, and it resolves over some time. In contrast, LPS purified from that asymptomatic E. coli does nothing at all. Okay, so we're able to, by purifying this LPS molecule, putting it into the bladder, kind of recapitulate the spectrum of clinical responses to infections with these different bugs. And I'm going to come back to this. You're going to see this image later on. All right. Now, how does LPS work? Well, LPS has a certain receptor on the surface of cells, and it falls into the class of what are called toll-like receptors. And that name comes from a gene first identified in Drosophila in fruit flies that encodes a receptor, an immune receptor. So this is an innate immune response. These toll-like receptors, or TLRs, mediate innate immune responses, not the ones immune responses that are generated as a result of vaccination, adaptive immunity, but innate hardwired responses that are usually associated with rapid inflammation. These toll-like receptors recognize conserved patterns of pathogens, including LPS, as we'll see in a minute. And usually a consequence of this is release of factors that stimulate immune responses and draw in other white blood cells, such as cytokines. And the receptor for LPS is called toll-like receptor 4, TLR4. And this was resulted in the 2011 Nobel Prize for Bruce Beutler. It gets very complicated what happens when LPS, as I've kind of sketched here, interacts with the toll-like receptor. First of all, there are a lot of players in this interaction. There are players at the cell surface that bring all these components together. There are a lot of important adapter molecules, and then these cause certain genes to be turned on, often genes involved in inflammation. And it turns out that TLR4, like I said, is the receptor for LPS. So here's looking at pain in mice. This is normal. These are normal mice infected with our uropathogen. When we look at similar mice that are lacking the toll-like receptor, they have reduced pain responses. Their inflammatory response, this is a marker of inflammation, myeloperoxidase, is comparable in response to both our pathogen and our ASB E. coli, LPSs. All right. So what that told us can be summarized here. And just looking at this right half, we find that LPS can do two things, and they seem to be separable. And this is kind of like the mast cell story with histamine and TNF, is that, that LPS can cause pain, but it seems to be distinct from inflammation. So two different LPSs that have come from bugs with very different pain phenotypes have comparable inflammation, but nonetheless very different pain when put into the bladder. All right, so that shows us that LPS can modulate the pain phenotype or define the pain phenotype. And where does that modulation come from? Well, as I mentioned, here's our schematic of this lipopolysaccharide molecule. Here's the lipid part, the fatty part. And then we have this big carbohydrate structure, the O antigen, and it's not really known what that does. Well, what we found is that different O antigens can modify the pain response of bacteria. So if we have bacteria, in this case, a very benign bacteria with O antigen, they cause no pain when put into a mouse bladder. But when we strip off that O antigen, now that LPS lacking this carbohydrate structure, these bacteria cause pain that persists weeks and months when put into mice. In fact, the infection is cleared very rapidly in these mice because there's a very benign E. coli strain, but nonetheless, a single infection with a bad bug, if you will, can cause chronic pelvic pain in mice. Okay. Let's see. Okay, so using approaches like this, we're able to study some of the signaling that happens somewhat peripherally, but initiated in the bladder by a bacterial infection with E. coli. So as I mentioned, LPS can interact with this toll-like receptor 4, and that's necessary for the pain response. And we have yet to complete the kind of complicated genetic experiments that tell us where this TLR4 is, whether it's on the urothelium that lines the bladder or on the end of these sensory nerve fibers. But nonetheless, we know TLR4 is important for pain, as is TRPV1. TRPV1 is a molecule that is known as the receptor for capsaicin, the active ingredient in red pepper. All right. We found that TRPV1 is required for these pain responses, particularly in chronic pain, for the establishment of pain. But if you treat TRPV1 as a target later, it has no effects. So it's necessary for the establishment, but not the maintenance of chronic pain. In contrast, there's a receptor CCR2 found on sensory neurons, and its ligand MCP1, which is elevated in certain pelvic pain conditions, most noticeably in men with prostatitis, and that this interaction is necessary for the maintenance of chronic pain. So we're beginning to understand some of the ways in which E. coli or various bacteria in the bladder can trigger chronic pain. And understanding that bacteria can modulate pain led us to think about other sources of bacteria besides an acute urinary tract infection. And many of you will have heard of the microbiome. So this is the population of bacteria and fungi and viruses that occupy any biological niche. Soils have a very rich microbiome. So does, of course, your gut. So stool is rich in a microbiome, as is your skin, your mouth, excuse me. And so these are all sources of microbiome. And we were thinking, well, we know that bacteria can modulate pain responses. Could we have modulation of bladder pain responses in IC by a microbiome? And if so, where might that reside? So we looked at the vaginal microbiome versus the gut microbiome of IC patients versus controls. And we started this with a pilot group of patients, and it became very clear that there was no association between the vaginal microbiome and IC, at least in our small cohort, but we saw an association with the fecal microbiome. All right. And so we studied that further and looked at the fecal microbiome of IC patients versus controls. And this is a breakdown of large classes of bacteria shown here. And some of them, there's very little difference, all right, between IC and control patients. However, in some of these, we can see that there's a huge expansion of, some classes of bacteria, all right, in IC patients relative to controls. And in other ones, here's one where there's or a couple of them here where there's actually a deficiency of these classes of bacteria in IC patients relative to controls. And we actually looked at this a little more closely using very quantitative approaches. And we found that there were several of these specific actual species of bacteria, both, gram negatives and gram positives, that were deficient in IC pelvic pain. We call them DIP species, deficient in IC pelvic pain. And some of these here, when we did careful quantitative studies, could be used as potential biomarkers, stool-based biomarkers for IC. So we think that these might be developed eventually. It's not ready for prime time, but might be developed as stool-based biomarkers for IC. We also found that among these, that they actually form an apparent community, so they co-vary. All right. We also looked at some of the metabolic features of these populations, and we found implications for lipid signaling. And some of you may have heard that short chain fatty acids are mediators of effects of the microbiome, and that seems to be the case with our samples as well. So that's in IC, that's in patients, but it also got us thinking, of course, about mice. All right. And I'm going to backtrack for just a second here and tell you about a little bit about mouse genetics. And this is using that original chickenpox virus model, comparing the two most commonly used strains of mice in biological science, the C57 black 6 mouse, or B6, and then the BALB-C mouse. And when we infected those in our acute model of IC with our chickenpox virus, we saw a big difference in the amount of pain. So these mice here suffered a lot more pelvic allodynia than our B6 mice did. We used this along with a semi-complicated genetic approach that I'm not going to describe, but to identify regions of the mouse genome that were implicated in this pelvic pain sensitivity. And we found one region here on chromosome 13. And that led us to identify a gene with a very complicated name. It encodes a protein called acyl oxyacyl hydrolase, or AOAH. So we've spent a lot of time looking at AOAH since that discovery. We found that it's expressed from the bladder to the spinal cord, where the sensory inputs come in, to key regions controlling bladder function in the brain. Such as the Barrington's nucleus, as it's called in rats, and mice. In humans, this is often called the pontine micturition center. And an example of the role that this can play in pelvic pain is shown over here in a couple pieces of data. So here is that VMR. We're measuring the response to increasing pressure in the bladder of normal mice. And our mice lacking this AOH gene are very sensitive to this. They have a much greater response. So their bladder is hypersensitive. So you can think of this gene as perhaps suppressing pelvic pain. When we give them an E. coli infection and look at chronic pain, again, these same mice have a much more dramatic response compared to what is, on most scales, this is about a 200% increase. This is big, but it's much bigger in these mice. So this is one of our IC models, these AOH mice. And there's a lot that's interesting about this mouse. I was fortunate to see a talk by Rita Valentino, and she looks at the role of a certain factor, corticotropin releasing factor, and I'll tell you what that does in a minute, and its role in bladder control and stress responses. And we had just done the staining in the brain for this AOH protein to figure out where it was expressed, and it was expressed in different circuits that she implicated in stress responses and bladder control. And interestingly, this is the first publication of brain imaging in pelvic pain patients. It happens to be from men with prostatitis, and in this paper, they were most interested in effects in the cortex. But what we note here is this region here that clearly lit up, and this corresponds to the Barrington's nucleus where we find AOH expressed. So we thought that was another clue as to what's happening. So why is this, this CRF, corticotropin releasing factor, so important? As it turns out to be in our models too. Well, it turns out it is at the very top of the hypothalamic pituitary adrenal axis that goes through. There's a normal daily rhythm to this, and it also mediates your flight or fight response as well as many other things, including mood. All right. So a part very nearby these circuits that we've been talking about within the hypothalamus releases CRF. This acts on the pituitary, as the name would suggest. Acting then on the adrenals, and then which then in turn produce glucocorticoids, and then that feeds back to shut this cycle down. And it turns out that alterations in CRF signaling are associated with changes in mood, and particularly depression and anxiety. And it was also shown by Rita Valentino's group that when you turn this on inappropriately in the voiding centers of the rodent brain, that you get altered stress behaviors as well as altered sphincter tone. This is a little different in rodents from humans. But as a result of all this, this is what I showed you about IC. Our AOH mouse is very similar. It does almost all of this perfectly. The one caveat here is that because the CRF controls sphincter tone so exquisitely in rodents, that these rodents, rather than urinating more, actually urinate less. That's the one problem with this model. But nonetheless, it recapitulates many other aspects of this. Of course, these mice get chronic pelvic pain. They get rodent correlates of anxiety and depression. There's a role for mast cells. They have leaky urothelium, HPA axis dysfunction. They also have perhaps gut dysbiosis, and that came to us when my postdoc was dissecting these mice, and this is the GI tract of a mouse here, and here's a normal mouse, and this structure here is called the cecum. This is very small in humans, but it's large in a rodent. But you can see in an AOH deficient mouse, it's much bigger, and I had no idea what that meant when Bin showed me this. So of course I Googled it, and it turns out that an enlarged cecum is a feature of germ-free rodents. Rodents grown in a germ-free environment such that they don't have a microbiome. And that suggested that there might be a role for the microbiome in these mice. And this is one way to show that. This is called a PCA plot, or a principal component analysis. If you look at some microbiome literature, sometimes you'll see plots like this quite often. And in a plot like this, what we're showing is this is 2 dimensions in 3-dimensional space, where a point represents a whole population. So think of comparing a Brazilian rainforest with a forest in upstate New York, and reflecting all of that diversity in the flora and fauna as one point in space. All right, and what we can see from this is here's the microbiome of normal mice versus our AOH mice with the big cecum, and they do indeed have very different microbiota. Now, it can converge. So it turns out when mice are in a cage together, they actually eat each other's fecal pellets, if you can believe that. It's called caprophagy. And what you can see is by co-housing the mice that their microbiota converge. All right, so you can modulate their microbiota. All right, so what does that do? Well, this is just to further emphasize that there are differences in microbiota. We can look at individual species. We can look at phyla. Some of the major phyla are not much different, but when you look at the minority one, they're very different between normal mice and our AOH mice. But it turns out that the microbiota can actually shape the pelvic pain responses. So we did an experiment where we took some stool of IC patients versus controls, and we cultured up the bacteria from those stool samples, and we gave it to the mice, and then we measured their allodynia. And the bacteria of the healthy flora, if you will, from the stool of normal controls actually seemed to cause the pain to go down, whereas the pain went up when they were given cultures from IC patients. So that suggests that the microbiome might play a role in modulating pelvic pain in humans, or at least those bacteria may have the ability to do that. Now, how can that happen? How can events in the gut modulate the pain responses? Well, that brings me to a certain kind of cell that's prevalent in the central nervous system called microglia. All right. So microglia they represent about 10% of the cells in the central nervous system, the CNS. And these are actually like a specific kind of white blood cell leukocyte called a macrophage. So these are macrophage-like cells that are innate immune cells. These ones happen to be resident in the CNS. And as innate immune cells, they actually express those toll-like receptors, so they respond to features of pathogens. Now, what is the relevance of that? Well, it turns out, and many of you may have heard this in the lay press, is that often when you have altered microbiota in the gut, that you actually get leakage from the gut. All right. So this leakage can be of a variety of things. It can be metabolites from these bacteria, and it can also literally be parts of the bacteria, such as LPS. So if you have a leaky gut, these things can go and act on toll-like receptors throughout the body inappropriately. So if they're triggering toll-like receptors in the liver, they can alter metabolism and drive obesity. If they're altering toll-like receptor responses in the microglia, or they're triggering microglial activation, these microglia then can release inflammatory factors such as what we call cytokines that alter neuronal responses. And it's been shown that microglia, when they're activated, release factors that can alter pain processing. So this is a very short summary of a paper we published recently and a couple papers in the works. And what we find in our AOH model, and also in another entirely unrelated IC model where we induce chronic pain with a one-time urinary tract infection, that we do have a leaky gut. All right. And this leaky gut causes, microglial activation. It's dependent upon toll-like receptor 4, and this is required for pain modulation. So for example, if we take our AOH mice and we silence microglia, their pain drops significantly. All right. And these effects, as I say, are mediated by these toll-like receptors. So we have leaky gut here and features of the microbiome causing activation of microglia, and these play a significant role in pain. All right. So I've told you a lot of bad news about how bacteria cause pain, but we do believe there is some good news. Again, not quite ready for prime time, but hopefully something we can develop. So I showed you that we can take mice and co-house them and alter their microbiome. And this is pain testing in those mice. This is that VMR. And here are AOH mice that are more sensitive than their normal mice. So these are mice lacking the AOH gene, and normal mice are much less responsive. So these mice have pain. The co-housed mice, they have reduced pain. So remember, their microbiome is altered. It's much like what you might have heard for C. diff colitis, fecal microbiome transplant, FMT. That is able to resolve the pelvic pain in these mice. Similarly here, we wanted to confirm that it wasn't just an artifact of socialization by putting normal mice with our pelvic pain mice. We gave them stool slurries for a week, and we saw the same thing. We're able to drive down the pain of our mice that are our IC model, all right? And I'm not showing it here, but we were able to also cure the depression of these mice through this treatment, all right? So an IC, a frequent IC comorbidity, resolved their anxiety and depression, as well as their pelvic pain, by modifying their microbiome. All right, so the microbiome is a way to modulate pelvic pain. I want to just return for a moment to this that I showed you before, where our pathogenic LPS caused pain when put into the bladder, whereas this one did not. And it caused us to think, is there actually something happening here that we're not seeing? And so we looked at the effects in acute UTI. So we're infecting mice with E. coli and giving them a bladder infection. They get pain, it resolves. And this is when we give 3 installations of saline as a therapy, starting 1, 2, 3, has no effect on the pain. When we give ciprofloxacin, the standard antibiotic for UTI, it also does not affect the pain. It's because the pain stimulus has already happened, all right? It doesn't mean mice, you can't treat their pain, because if we put 2% lidocaine into the bladder at the peak of their pain, it drops it by about 70%. But if we give a single intravesical dose of our ASB E. coli into the bladder, it really knocks down the pain, all right? Turns out that looking at a whole panel of these different asymptomatic E. coli strains, that some of them are much better than others. Some have very nominal effects on pain, but some are very significant. And it works not just when put into the bladder. So here's our untreated pain. We put it into the bladder, it knocks down pain. We can put it into the vagina or the colon, suggesting that we could potentially develop a therapy like this as a vaginal probiotic, or even a colonic, an oral probiotic, meeting its effect through the gut. And it doesn't just work in chronic pain, it also works, or in acute pain, it works in chronic pain. So it's kind of backwards from what I've been showing you. This is now a pain relief scale and analgesia scale, because these mice have been in pain for months, or weeks to months. And saline has no effect. It confers no analgesia, but when we give a single dose of our good E. coli, if you will, we get a benefit that lasts, in this case, about 3 weeks, all right? So we believe that this is another way in which we can modulate pain. So just to kind of wrap this up, I've talked about how, pain signaling, we'll just leave that there. Pain signaling is mediated by receptors, particularly these toll-like receptors, and it's defined, the pain phenotype, by these LPSs, and some are bad and cause pain, some are good and are associated with analgesia, and we can use E. coli or even purified LPS to block this pain signaling. And even, I didn't talk much about the spinal cord, but seemingly reverse events in the spinal cord. In terms of the etiology of IC, it also, this whole model here suggests that perhaps if an unfortunate patient gets a UTI, a one-time UTI, or a recurrence with a wrong bug, a bad bug, and they have the wrong genotype, for at least a subset of patients, this might result in chronic pain. But perhaps we can use E. coli to treat this. I'm not going to talk about that. But remember, we have these LPSs that we know are really the drivers of the pain phenotype, some of which are analgesic. And this is a story that's emerging in our group. We haven't published anything yet, but we're starting to actually work with a colleague where we get away from the whole bug and we present this beneficial, this analgesic LPS on the surface of a nanoparticle. And we find that these also are analgesic. So kind of tying all of this together, we think that we can take these LPSs, and whether presented in the context of a new high-tech nanoparticle or on the surface of E. coli, either put into the bladder directly or perhaps via the gut, that we can use crosstalk or direct instillation to modulate pain. So with that, I thank you very much for listening to this talk about the role of bacteria in E. coli. Of course, many people were involved in this work. Most notably, I want to mention Tony Schaefer, a renowned urologist who I've been working with for over 2 decades now, numerous collaborators, as well as generous support from the NIDDK. And with that, I would be happy to answer any questions if there are any from the audience. All right, I'll just leave that up as a summary. Let's see. Okay, someone wants to know about this presentation. Sure, I'm happy to share this. Probably more comfortable sharing it as a PDF, if you don't mind. But yes, I'm happy to share this. And I believe that the recording of this conversation is going to be available online. What treatments are commonly available? So I think many of you will know this, and if you're a recent IC patient, unfortunately, we don't have a lot of treatments that are available. And I'm not a clinician. Again, I want to emphasize that. There's one FDA-approved therapy, Elmeron. It works in a subset of patients. I believe the numbers are about 25% of patients respond well to Elmeron. Dietary management is really a classic first thing that many patients will find that, as we mentioned, comestibles are triggers that you would want to avoid. And I was talking with a urologist once who told me he had a patient who she found that her only trigger was strawberries. All right, and when she realized that and was able to eliminate strawberries entirely from her diet, that she found great benefit from that. So classically, we hear about acidic foods and we hear about caffeinated foods, but there are other triggers. So unfortunately, that is a lot of trial and error that is required. So here someone writes, Jennifer, so you're suggesting that at some point stool slurry of good E. coli could be a treatment for humans. Yeah, there are a lot of ways to approach this. Perhaps taking this ASB E. coli itself might be sufficient, or it would be a reach to say a fecal microbiome transplant in patients, but that's what we find in mice works. Indeed, manipulating the microbiome in mice works. In fact, giving whole stool slurries works, or them through caprophagy ingesting the fecal pellets. So that might be a future therapy, and we know that from the microbiome press and literature that this is being employed successfully to treat other conditions. Let's see. Well, I'm glad. A Zoom user liked the presentation. Thank you. Can anti-inflammatories help? That's a good question. So what would the anti-inflammatories be? NSAIDs, I haven't heard anything, and I would defer to maybe patients who have tried these. I haven't heard much about success using anti-inflammatories, NSAIDs, for example, like an ibuprofen, naproxen, kind of treatment providing much benefit. That doesn't mean that it couldn't be tried. You know, I will tell you this is an N of 1. So you know, at your own risk, my mom found as a result of our conversations that some antihistamines provided benefit to her. And she tried Benadryl, which provided some benefit. She got better results with ranitidine, and maybe that's because that's what her son told her to try. You know, because we found in our mice that the best antihistamine was ranitidine. That was the most effective, and that targets H2. I have a colleague who does very similar work, and he is trying to develop a therapy based on blocking H1 receptors, very similar kind of thinking, and in male mice, it was the H1. Oh, okay. Benadryl, so someone is saying, Jennifer is saying anti-inflammatories, she's not found them helpful. Benadryl provided some relief. Hydroxyzine, you know, it's kind of interesting within the urology literature, there was a clinical trial of hydroxyzine, and it was not found to be effective. This is a very early antihistamine. And so there are a couple reasons why it might not have been demonstrated to be clinically efficacious. Hydroxyzine, first of all, it's an early generation one. There are more potent antihistamines that are available now, including those that target H1 and H2 specifically. Hydroxyzine is more of a blunt tool, but also, unfortunately, you know, a lot of drugs, and that's why, you know, well, pain is notoriously difficult to treat in the form of a clinical trial because the placebo effect can be so significant. You know, from 25 to 40% of placebo effect has been observed in pain studies and clinical trials. So that makes it difficult statistically to even show a benefit. If you may be getting actually a modest benefit, but it may not appear clinically significant, you know, at the level of statistical tests. But good for you if hydroxyzine is helping. Okay, PDF, Janet's happy with. Carolyn, any thoughts on H. pylori and links with Hunter's lesions? I have not heard of that. For those of you not familiar with H. pylori is a pathogen that causes gastric ulcers. And so that's why gastric ulcers can now often be treated with antibiotics. I am unfamiliar with its links to Hunter's ulcers, but that's a very interesting idea. The one thing about that is, you know, many of us are familiar with the term of IC being a diagnosis of exclusion, right? And one of the things that on the NIH, NIDDK research definition for excluding conditions that could give rise to IC symptoms is excluding the possibility of UTI. So often patients will go through a series of antibiotic treatments to eliminate the possibility of UTIs on the way to their IC diagnosis. So there may be something in the literature I'm unaware of with H. pylori, so I will go and do some homework on that myself. Oh, and so we were looking at what we were comparing in our study. Ashley asks about the role of the vaginal microbiome and whether that might be linked to IC. So the way we structured we're working with collaborators at the University of Illinois at Urbana-Champaign who were computational experts. And so what we did was we took our first 10 patients or so in each cohort, or in healthy controls versus IC patients, and we compared the vaginal microbiota with the fecal microbiota and their symptom scores. And we found that their symptom scores varied with bacteria in their fecal microbiota, but not their vaginal microbiota. So that's why we began to study in earnest the fecal microbiome. Let's see. Do they have a history of vaginal infections? Not that I recall, no. In fact, bacterial vaginosis would have probably been an exclusionary criterion for that study. I know it was. Let's see, Janet asks, has been suggested that bacteria within the bladder may be addressed with the use of low-dose antibiotics. Your thoughts? Prophylactic antibiotics. Yeah, it turns out that probably the world's foremost expert on this, who actually did a lot of his early training here at Northwestern, is Scott Hultgren. He's at WashU at St. Louis, and he's probably the world's foremost expert on urinary tract infections. And he has for probably decades now shown that bacteria, E. coli particularly, can reside within urothelial cells or within the bladder tissue. And he has tried various strategies. Probably the one that I think has gained the most traction, at least from the research and drug development front, is using low-dose antibiotics in combination with sugar mimetics that block adherence of E. coli to the bladder cells, and that shows some promise. Let's see, Keflex is doxycycline versus Keflex. That's beyond my area of expertise. Let's see, quercetin, patient by the name of iPad, from her iPad says that quercetin helped me significantly, and I think that Dr. Theoharides champions that as well. So I'm happy to hear that provided you with benefit. Erica, do you believe that there could be a biofilm covering the surface of the bladder and making them not targetable? So this is really alludes to the work of Scott Hultgren, again at WashU at St. Louis, and we've done some of this ourselves as well here at Northwestern. And yes, he has shown that both in mice, as we have, and then gone on to show that within humans that there's an entire intracellular life cycle of E. coli and other uropathogens within the urothelium, within the bladder lining cells. So not really atop the bladder, in the lumen or on top, but within the bladder lining tissue itself, the urothelium or the urothelial cells. And that, oftentimes they're forming biofilms. And for those of you not familiar with biofilms, these can be dense aggregates of protein, of bacteria that can exist on surfaces or apparently within bladder cells. And they can be refractory to antibiotics when they're in this biofilm state. They can be at times in a state of suspended animation where they're not susceptible to antibiotic treatment. So yes, good point there. Janet, mast cell inhibitors such as Pepsid, cromolyn and other blockers. Yes, so the mast cell inhibitors, again, Pepsid, I don't know, I'm not really a pharmacologist or a drug person per se. So I don't know which one Pepsid is. I believe, I shouldn't speak about Pepsid. Cromolyn, though, cromolyn is a mast cell inhibitor, and it's a very good point. In fact, my colleague who studies prostatitis is trying to develop a therapy based on a combination of cromolyn with an antihistamine. All right, so a very good suggestion there. Shelley says that hydroxyzine helped her a great deal. Someone writes in, would you recommend DRG implants? So the dorsal root ganglia, these are they run along the spinal cord, and this is where the sensory nerve fibers aggregate. The dorsal root ganglia, they're kind of like nodules running along the side of of the spinal cord, and then that's where the input goes into the dorsal horn, as it said, the tops of the spinal cord. Well, there are people who receive benefit from sacral implants that stimulate nerve responses, and so there's a lot of literature on that. I personally would, I can't speak to that very much myself. I've worked with physical therapists. All right. It's interesting because physical therapy, pelvic floor physical therapy, has been shown to statistical significance in a clinical trial to benefit IC patients. So if you can find a pelvic floor physical therapist in your area and you haven't explored that option, that might be, that's been shown previously in a clinical trial to be efficacious. Support groups, great participants. Support groups, yes, and the ICA, hopefully the ICA is helping you to form a greater community and find these management exercises. This person mentions yoga, Tai Chi, relaxation, and as I kind of mentioned, and it's here on this response or on this overview, is that corticotropin releasing factor, which is part of it's the initiator of the HPA axis, which mediates stress responses. It is also well known to modulate pain responses. So anything you can do in the area of yoga, Tai Chi to reduce your stress, and I think that it's been well documented and it's well known amongst patients that stressors can cause flares. So anything you can do to reduce your stress may help with your symptoms. Desert harvest aloe. Each person is unique. I think that says it very well. You have to find your own pathway to effective management of your condition. Some rotate probiotics. This is something that, because probiotics, am I a fan of probiotics? And I'm just going to offer this generally. Yes and no. As you saw, we tested a panel of some 24 different ASB strains to find those that are most analgesic when put into the bladders of mice. I wish that was done with all probiotics. In some probiotics, there actually is a big literature, but in others, there isn't much of a literature. They took it because it's a normal constituent of stool, but that doesn't mean that it's an important player necessarily, even if it's very abundant. We're beginning to understand that there are certain keystone bacterial species that are necessary they play key roles in forming these critical communities of various species. They play key roles in metabolism of certain factors. It's not clear that probiotics necessarily play this role. They may be there normally, but that doesn't mean that they're necessarily going to be, just because they're a commercial product doesn't mean they're the most effective at doing the job that we hope they'll do. There was even a study that came out a few years ago that they did both in mice and humans, where they treated patients with antibiotics, and then they either gave them autologous, so self-stool transplant, or they gave them probiotics, or they gave them nothing. And they looked at how the microbiota recovered. Obviously, the patients and the mice receiving self-stool transplant, their microbiota immediately returned to normal or very rapidly. The people given antibiotics and mice and allowed to recover naturally got most of the way there. The people and the mice treated with a cocktail of, I believe, 12 probiotics did not return to normal during the course of the study. So probiotics, I think, are maybe worth trying, but especially if you find someone else who's tried a particular probiotic, I would follow their lead. Any thought on tricyclic antidepressants? Well, apparently tricyclic antidepressants, besides being antidepressants by name, also play a role in can actually help to silence mast cells. So that might be one mechanism by which they work in IC. Many patients use tricyclics for managing IC. I know my mom did for a number of years. Connection between mast cell activation syndrome and IC. I apologize, I'm not familiar with MCAS. Kevin asked that question. Any evidence that viruses, in addition to bacteria, are drivers of IC? I don't know that there's any evidence, although I find it interesting, that we use a chickenpox-like virus, PRV, which is most similar to zoster, right, which causes shingles, and we get basically what I like to call shingles-like condition of the bladder in mice. I've long had an interest in that possibility, but there's nothing definitive yet. Erica asks about the biofilm. That's Scott Hultgren, Hultgren. He's a member of the National Academy of Sciences, very highly regarded scientist, and he's at Washington University of St. Louis, and he's the world's foremost authority on urinary tract infections. Need to find the study for my urologist. There's a dense literature from the Hultgren lab, who, again, put in a plug for Northwestern. He started here. Janet Warren, agree with Erica, biofilm therapies, yes. Ashley, stress also lowers biodiversity of the gut biome, correct. And there are a lot of, yes, I'll leave it at that. Maureen, you're welcome, Maureen. Janet, all of our health studies is coming on the high number of links I see having with lichen sclerosis. And I don't know much about lichen sclerosis, so I don't know about the links, but there are a number of comorbid conditions with IC. All right, I'll put together with the PDF, I will add some of the names. Okay. Recording, and as the ICA is saying, we will be providing the recordings. With that, I will thank you again for joining this presentation. I hope it was interesting and somewhat informative, and I will hand this back to Jenlene to close.
Thank you so much for being here with us today, Dr. Klumpp. This session was very informative. If you found this event to be of value, please consider joining us for future webinars and share with your community. We are also excited to announce next month's webinar with Dr. Robert Moldwin. He will be talking about the pelvic floor muscles, another possible source of pain for the IC-BPS patient. So watch your email for more information. Thank you so much for joining us today.
Wonderful.